Good evening, everybody. I'm going to start off by just saying that as you all look at me here, vision is the most precious of our senses. And we have learned so much over the last years about how light goes into the eye, gets processed in the retina, in the center of our vision, the rods and cones, the photoreceptors, gets collated and goes down the optic nerve head, passes out of the eye, gets carried back to the brain. We've learned so much about how vision works when it's working right. We also know a lot about how it goes wrong, the unmet needs in vision care right now. A lot of unmet need when we look around the world is the need to distribute glasses or deliver cataract surgery to places that can't get it. But heartbreakingly, so much of that unmet need is also irreversible vision loss diseases like glaucoma, age-related macular degeneration. These are among the most common neurodegenerative diseases in the world, in the United States, in our community. And we've learned a lot about what goes wrong in those diseases, taking those key first steps. We know, for example, in macular degeneration, we lose those rods and cones, the photoreceptors in the back of the eye, and their support cells. We know in glaucoma, we lose the retinal ganglion cells that gather up all that vision and send it down the optic nerve back to the brain. Developmentally, the eye is actually an outgrowth from the brain, and like the brain and spinal cord, there's no regeneration. So just like in a spinal cord injury, it leads to permanent paralysis. In these diseases, when we lose these cells, it leads to permanent vision loss. Luckily, in ophthalmology, it has historically been an enormously innovative field. So many firsts in medicine actually were brought to medicine by ophthalmology. The first tissue transplants, the clear window on the front of your eye, the cornea, the first tissue transplants in medicine were corneal transplants many years ago. The first use of lasers in medicine, actually done right here in Silicon Valley. The first use of lasers, also in ophthalmology for eye disease. The first FDA-approved artificial intelligence, AI. This now allows patients with diabetes to go to not just to our eye clinic, but any of our clinics and have a computer take a picture of their back of their eye and tell them, do they need to go into the eye clinic, get checked out, need therapy, or no, they're good, come back next year. Also, the first FDA-approved gene therapy can now be injected into the eye and restore vision in children and adults with hereditary eye diseases. We also have amazing firsts coming out of the laboratory. The first areas of the brain to be fully mapped so that we can start to see how all those neurons and cells are working together, in this case in the visual cortex, to process vision, to take all that data from our eyes and turn it into vision. Our faculty here at Stanford are leading in that basic science of discovery of how vision works. But we still have unmet needs in eye disease. We need better biomarkers. These are the ways that we take pictures or other measurements of eye disease. And this gives us ability to measure risk of disease, measure the diagnosis of the disease, measure who's progressing, be able to tell a patient before they get worse, I've got a measurement that says we've got to advance your therapy today and save your vision going forward. Of course, we also need better treatments to protect or even restore vision in these classically irreversible diseases. We use words like neuroprotection. How do we keep the cells from dying? Regeneration. How do we regrow the injured retina, the injured optic nerve? Neuroenhancement. That's a term where we take a sick but not yet dead neuron in our retina and we give it a booster shot so it can fire on all cylinders again and thereby restore vision in patients with these diseases. Of course, our patients come in and they ask me, well, I've got bad vision. Can't you just do an eye transplant? For the longest time, this hasn't really even been on the table. But actually, for the first time now, it is on the table. And to attack this problem, we've assembled a collaboration of 48 faculty at Stanford and at 23 institutions, collaborating institutions around the country, to go after total human eye allotransplantation, whole eye transplant. Thea is also the Greek Titan goddess of vision, so that fits. This has a lot of challenges associated with it. There's a lot of science that's going to have to go into it. We've got to go after the viability of the tissues. We've got to think about how to image, figure out who's a good donor. Are we keeping the tissues alive in between donor and host? The surgical approach, how do we take out the eye? How do we put back in the eye? Immunomodulation, we've got to control the immune system so it doesn't reject. Ocular preservation, keeping that eye alive in between donor and patient recipient. And of course, the hardest problem, neuroregeneration, getting those tissues, getting that optic nerve to regrow back to the brain. And you put that together and it's vision for Thea, because you can't have an ambitious project without a good acronym. So we're now going after this, and we are making amazing progress already just our first year into this ambitious program. How to identify the best donor eyes that are the most healthy and most likely to succeed in an eye transplant. How to surgically explant and preserve the tissue properly. How to keep the eye alive, perfused, living and healthy in between donor and host, recipient, patient. Of course, how to implant it surgically back into the patient. How to block the immune system. Do we have to block the immune system in the entire body? Or could we just block it up there at the eye and save the patient a lot of systemic disease? We're making advances on all of these fronts, local delivery, surgical approach, every piece of this team is now making innovative advances. And of course, the hardest part of this problem is how to regenerate the optic nerve back to the brain. But what we're seeing here is actually an amazing picture. One of our Stanford faculty in this collaborative program and his laboratory has actually figured out a cocktail of 5 gene therapies that can regenerate in our preclinical models fibers from the eye all the way back to the proper spots in the brain. This is another big first. We've put together this amazing collaboration that's just a joy to work with. We have faculty at Stanford who are the surgeons figuring out how to innovate in the surgical approach. We have faculty at Stanford who are the world leaders in how to do the imaging, to measure the eye, biomaterials, how to knit the optic nerve back together, and of course, the biggest problem we need the most people to work on, the faculty collaborating to work on regenerating the optic nerve. Just as amazing is the opportunity to work with all of the trainees and laboratory staff who work every day to make this all happen. Now, on the way to this ambitious goal, we're also trying to already today take these advances out of the laboratory and into the clinic and have like kind of the side spin out advances that could help our patients even today, even before an eye transplant. So how are we doing that? We're doing clinical trials at Stanford that are letting us lead the way. For example, we discovered years and years ago that there are protein growth factors that keep the neurons alive. So we've moved that from animals into humans. This was a study we did here, testing nerve growth factor eye drops and showing in the green part of the circle that we could actually improve the retinal anatomy that's normally lost in glaucoma. Even more exciting, we've just finished a trial using insulin eye drops, where you can see here on the right the improvement in retinal thickness that's normally lost in glaucoma. Another growth factor that we can deliver with an implant is called CNTF. And here, in the rightmost bar set of this bar graph, we actually found that you can basically halt progression in patients with glaucoma, keep them from progressing altogether. Really an amazing advance. This actually was just FDA approved for a different retinal disease called macular telangiectasia, and we can now make it available to more patients at the Eye Institute. We learned that actually stimulating with electrical activity can increase the responsiveness to these growth factors. And we're testing in clinical trials now both visual stimulation and very exciting electrical stimulation. And in our patients over at the eye clinic, for the first time, we're seeing improvement in their vision after these treatments. We're testing metabolic approaches like hyperbaric oxygen, high-dose vitamin B3 in carefully controlled clinical trials. Putting this together, I couldn't be more excited about what's right ahead of us, the progress that we've been making, the progress that we're making, as we move these advances from the lab into the clinic, from the science into the patient, and really see this as an opportunity to restore vision, protect vision in our patients. And I thank all of you for helping support and accelerate all of this work. Thank you.